Good evening, everyone. I hope I am audible to every one of you. And you can see my screen as well. Please respond in the chat. I'm clearly audible and visible. Okay. So I hope everyone is okay, fine. So I could see Sakshi, Nandini. Okay, good evening. Yes, good evening, everyone. Uh, Kritika, good evening, everyone. A very good evening and a warm welcome to the day 5 of RRR session. I hope you are really getting benefited with the previous sessions taken up by our eminent faculties of Biotecnika. And today's session is regarding Unit 6 and 7. Okay, so without wasting much time, students, let's start with our session. Okay, so first we will be taking up Unit 6. And that is, we all know what is Unit 6 of your CSIR syllabus, and it's System Physiology Plants. Okay, so let's start, students. I hope everyone is ready. So give me a, no, please type in bio in the chat box. Let me see. Let me see. I want to see. Bring it on, please. If you are ready and you are giving me a go ahead, then please write, bring it on. Wow, that's great. That's great. So I could see a lot of bios coming up in the chat. Thank you so much, everyone. Thank you so much. So this gives me that energy to start the webinar, right? Okay, that's great. So let's start with our first question of Unit 6. Yeah, thank you so much, everyone. The first question is, following are certain statements regarding plant respiration. The first statement says, and you have to first give me the answer. You have to try. So I'll not be showing you the answer directly. You have to try, and then definitely I'll be explaining you the reason. Okay. Now, so metabolism of glucose into pyruvate through glycolysis generates NADH and not NADPH. Metabolism of glucose through oxidative pentose phosphate cycle does not produce NADPH. Cyanide forms a complex with heme iron of cytochrome oxidase, leading to prevention in the change in which valency, which in turn stops electron transport in respiratory chain. And the last one is alternative oxidase is insensitive to cyanide and has higher Km. So I could see already the answers are coming in. So please, let's try. And definitely you could very well understand from which part of your Unit 6 syllabus this question has been given. So it's from 6B, that is plant respiration, right? So everyone is saying 4. Okay. Okay. Let's disclose then. Yes, a lot of you are saying 4. Absolutely correct. The answer is, the correct answer combination is 4. So see, I have students kept this glycolysis no slide and this image in front of you. So let's go back to the question first, how to analyze. So 4 is option combinations A, C, D. Okay, this question came in a very recent CSIR examination. So metabolism of glucose into pyruvate through glycolysis generate NADH and not NADPH. Absolutely. So if you just go back to the pathway, and if you can see in this step, students, that is glyceraldehyde 3-phosphate getting converted to 1,3-bisphosphoglycerate. I hope it's visible to everyone. So here we could see that 2 molecules of NAD plus is getting converted to 2 NADH. And what this step can be called as? Any guesses, students? What should we call this step to be as? Let me see what this step should be called as. Like, this is what should be the step name, students? Come on, you will be able to say this. When NAD plus get converted to NADH, it's called as oxidation, students. Oxidation. Why? Because NAD plus is itself an oxidizing agent, so it oxidizes the substrate and it gets reduced itself. So for this reason, we call such step to be as oxidation. Very good. Aditi has given an answer. Very good. So the first option, statement A, is correct. Next, metabolism of glucose through oxidative pentose phosphate cycle does not produce NADPH. Yes, definitely. So when we will be doing pentose phosphate pathway, or if you have done pentose phosphate pathway, we have seen also that it's being told that it does not produce NADP but NADPH. So it's a wrong statement, because in pentose phosphate pathway or hexose monophosphate shunt, NADPH production is a must. Okay, so that is a very important observation that we have to follow. In glycolysis, we are not getting NADH, but in pentose phosphate, we are getting NADPH. Correct. So B is an incorrect statement, so we are not taking in statement B. All right. So coming to C, so has to be, if B is incorrect, the other two options has to be correct by default. So cyanide forms a complex with heme iron of cytochrome oxidase, leading to prevention in the change of valency, and it stops. We know this, students. Okay, some of you I can see very common names. Okay, so you must have attended the classes, and it's well known to our students that cyanide, okay, it is a very important, when we talk about AOX, alternative oxidase, remember it's an uncoupling protein. So when the uncoupling protein in an electron transport chain will be activated, students, when there will be a cyanide stress, and cyanide will be binding to the complex 4 of mitochondrial electron transport chain. Correct. So absolutely C will be a correct option. Cyanide will bind to the cytochrome oxidase, and definitely it will hamper the respiratory chain action. Okay, so coming to statement D, alternative oxidase, we are all familiar with the name. So alternative oxidase is the another side. So this is the other side of the coin. The another side of the coin is called as UCP. We know this, uncoupling protein in animal system. Right? Yes, Sahiba, you're absolutely correct. It inhibits complex 4. Okay, and yes, and it has higher Michaelis-Menten constant value that of cytochrome oxidase. So you are absolutely correct, students. Combinations will be ACD. So let's move on to the next question. Just a second. Yeah. I hope you're able to see the question. Yes, now it has come. Yeah. Blue light receptor. Cry. So students, again, a very important question. It's important, but it's easy. So after, whenever we are taking up any question, I would be asking you that from which chapter it is. That is our first understanding that yes. So we are heading towards, the preparation is going right. We should at least know that from which chapter the question is coming. That is very important, students. All right. So tell me from which chapter is this question has come from. Can anyone tell me? Come on. Let me see. Come on, students, tell me from which chapter of Unit 6 this question has come. Let me see. I want some answers in the chat box. Yes? No answers? Okay, so Srinivas, some Shirin, I'm not, I guess I have spelled your name correctly. You have only mentioned sensory, but what is the full chapter name? Very good, Sahiba. It's sensory, it's photosensory inhibitors, but the chapter name is sensory photobiology. That is photoperiodism. Okay, so that is very, very important. Yes, Nupu, you're saying the topic, but I was asking about the chapter. So all of you guessed it correct almost. Now, so it is from Sensory Photobiology, Unit 6E, and it's a very important chapter, students. Okay. So it says that blue light receptor CRY1 binds to COP1 and SPA1 complex by interacting with C-terminal region of CRY1, CCT, in a light-dependent manner and regulates photomorphogenesis via transcription factor high 5. And now, based on this, we have some following statements given. Okay. Yes, Antara. So everybody can ping in your answers meanwhile, so we can analyze the questions, and then we can go to the answer checking. Correct. So when we say COP1 signaling, COP1 is a very important participant in sensory photobiology signaling. Okay, we know there is a very important signaling in the chapter that is called as COP1 signaling. What is the full form of COP1, students? Constitutive photomorphogenic 1. Okay, let me write the name of the gene name. If it's not constitutive photomorphogenic 1 gene. Okay, and what is SPA1 gene? What is SPA1? Suppressor of constance 1 gene. Okay, SPA1, suppressor of phi A gene. Suppressor of phi A gene. Very good, Mahima, very good. Suppressor of phi A is SPA1. So we all know, students, so what happens in COP1. So in my answer key, I have that pathway to show you all. So COP1 and SPA1, they are basically planning an attack, and that attack will lead to the inhibition of a particular form of photomorphogenesis, students, right? We have seen this. So after the SCOP1 and SPA1, they form a complex, they become more stronger, they become more powerful, and hence they inhibit that particular photomorphogenetic development. Okay, now here certain statements has been given, so just consider this to be as A, B, C, and D statements. Yeah, very good, Antara, very good, very nice. You remembered. So cry1, the statement said cry1 binds to spa1 and cop1 complex, leading to the degradation of high 5. Now students, I have to say something here. This high 5 is a type of transcription factor that leads to photomorphogenetic development, correct? So this is a transcription factor that bring about photomorphogenesis. Okay, and cry1 means yes, yes, Antara. So what is cry1? So cry is a photoreceptor that encodes blue light. Okay, so cry1 will be active in the morning time, and we all know that cop1, okay, and spa1, they form this complex during the night, and they mainly undergo ubiquitination of all the transcription factors during the night time. So okay, fine, so answer definitely is option 2. Some of you have given option 4, some of you have given option 1. No, dear students, the correct option combination, yes, some of you have definitely given the correct answer as well. So the correct answer is option 2. Okay, now, so why it is 2? So can you see this signaling mechanism? So it's written in the dark COP1, which is an E3 ligase enzyme. It will form a complex. Very good, Nida, very good. And they will be trying to ubiquitinate these 3 transcription factors. So there are not only 3 transcription factors, students. Along with high 5, we have LAF1, we have HFR1. So we have already done this in our class, if you have followed the class. And then what is happening during the daytime? This COP1 will be leaving from the cytosol. Okay, it will be leaving from the nucleus. I want to rectify this, and it will be moving out into the cytosol. But before it leaves, can you see that it is adding ubiquitin tags over phi A, and the transcription factors are becoming freed. Okay, so now they are ready to transcribe. That means cry1, when we are talking about cry1, students, cry1 is encoding blue light, and blue light will be prevalent. They will be active in the daytime, so they cannot bind to the cop1 ideally. Got it? So cry1 students cannot bind to spa1 and cop1 complex. No, they cannot do it. Okay, so statement A is incorrect. Now let's come to statement B. Cry1 binds to spa1 and cop1 complex, leading to, preventing the degradation of high 5. Yes, that is the correct option. Absolutely. Because cry1 is a positive regulator. It will be active during the daytime. Correct. So it will never help this 2. I would say they are the bad man. They are the bad persons. So they cannot allow SPA1 and COP1 to form complex, amalgamate, and undergo the degradation of the transcription factors. Okay, so cry1 will always try to prevent the degradation of the transcription factors. So absolutely, statement B is correct. Okay, now coming to the third statement, CCT. So CCT is another very important factor, and that is required for the entire regulation of the circadian rhythm, the biological clock of the plant system. So it has been mentioned here that by interacting with C terminal region of cry1, CCT. Okay, so CCT is the C terminal end of cry1 region. Okay, and definitely it regulates photomorphogenesis. So see, it is mainly overexpressed during the daytime. So but here unfortunately we have no combinations where both B and D is mentioned. Okay, so students, we have to go for only statement B, but yes, CCT is overexpressed mostly during the daytime. Okay, so that is why, as there is no chance or no spaces where we could find such option combinations that where B and D is there, so we have to go for statement or option B. All right, now I hope this is clear to everyone, students. So yes, Mahima has given some insights and light. COP1 movement towards outside cytosol is fast. And in dark COP1, yes, exactly so, Mahima. Exactly. Thank you, Mahima, for the feedback. Thank you so much, dear. So okay, Devanshi, thank you so much. So now we'll move on to our next question. Yes. I hope you're able to see the question. Debanjan, since there are contrasting statements, if we know between A and B, we know the answer. Yes, definitely. That is what. Because see, here it's very easy, Debanjan, to select the questions. Okay, so the previous type of question was very easy. Because see, yes, like the CCT expression, one of the statement was definitely correct, but we had no option combinations there to choose. Right? So that is the thing. See, right. So only members of the, now we are on the third question, students. Only members of the plant kingdom and many bacteria have the capability of biological nitrogen reduction. So in this regard, the following statements have been given. So it's very well evident that this question is from a very popular and an important chapter of Unit 6, and that is nitrogen metabolism. Okay, so let's find it out. What are the statements given here? Okay, statement A, nitrogen is normally taken up by the plant systems. Okay, already Nida and some of you have started giving the answers. Very good. Very nice to see your eagerness. Very nice. Very nice. So nitrogen is normally taken by the plants in their fully oxidized form, but needs to be reduced before the incorporation in inorganic molecules. Statement B says, conversion of oxidized nitrogen into reduced nitrogen needs energy in the form of NADP plus, and the metal associated with the enzymes nitrate reductase is magnesium. And the last statement, nitrate reduction take place in cytoplasm, whereas nitrite reduction take place in chloroplast matrix. Students, first and foremost, every time I say this in the class, and again I will be saying this in this webinar, that Unit 6, the most important thing about Unit 6 is remembering and memorizing the locations of different metabolic reactions. They are very, very important. Okay, we need to learn them very well. A lot of questions we have, or we have already discussed in our classes, or today also I will be telling you that locations, like when we talk about photosynthesis, there are a lot of things that we need to remember. Like there are a lot of pathways we have come across. So where do they taking place, where it's happening? See, you will get a lot of questions based on this, and we expect such questions in part C only, students. Okay, Now, so let me see, let me show you the answer. The correct option is option 4. So some of you have given option 3, okay? So most of you have given option 4. So option 4 is A and D. Now, students, the first thing, how to know, chop off the incorrect statements. That is very important. Now, is this correct? The metal associated with the enzyme nitrate reductase, repeatedly, many times I have mentioned in my class that this, the structure of enzyme is very, very important, students. You need to focus. Okay. And there only, see, we have got a question based on that concept here as well. Yes, very good, Nida. It's molybdenum. It's not magnesium, it's molybdenum. And along with molybdenum, we have another organic molecule we all know. I have the image with you all. See, MO is molybdenum, so it's not magnesium, students. Okay. And see, can you see the CO over here? So many times, what students know think is CO is cobalt, but no, it's not cobalt. It is complex, molybdenum complex. And why this complex is mentioned, students? The complex stands here for an organic molecule which is present along with molybdenum. It is called as pterin. So pterin is an organic molecule that forms a complex with molybdenum. Okay, yes, very good, Kritika, very good. And along with molybdenum complex, we have heme, and another one is we have FAD. So definitely it is not magnesium, so we can chop off statement C. Now, let's take up this. Okay, now we'll move on to the first statement. So nitrogen is normally taken up by the plants in their fully oxidized form, but needs to be reduced before incorporation. Absolutely. Whenever nitrogen, like see nitrate, nitrites, they are all oxidized form. But later point of time, they get reduced. So this is a very basic question. See, sometimes you know what happens, statement A, which has been given here, it's a very basic thing. It's a basic chemistry based question that you need to understand. Okay. So see, these are very basic, but you need to understand whenever you are doing. We are doing the chapter nitrogen metabolism, but we do not know that how the nitrogen is being captured or absorbed from the atmosphere. Then what point? So you need to know these things. So it's a very flat question. So definitely by default, statement A is correct. Coming to statement B, conversion of oxidized nitrogen into reduced nitrogen needs energy in the form of NADP plus. No, most of the time it requires in the form of NADPH. So if you have done the pathways, like there are 4 pathways of conversion of ammonia to amino acids, GDH, GS, GOGAT. So all these pathways are very important. If you go through thoroughly, you'll get to see the energy is not required in the form of NADP plus, but it is required in the form of NADPH. See, these are minute observations, but we have to do it very well with our full concentration. We cannot skip anything. And nitrogen metabolism is a very small chapter, but I would say it's full of information. So we need to learn, we need to do each and every topic from nitrogen metabolism thoroughly. Okay? Now, at the last statement, nitrate reduction take place in cytoplasm. We all know this because after nitrate, what do we get, students? Nitrite. And the nitrite reduction finally needs to be taking place in chloroplastid. Why? Because nitrite will reduce to give us ammonia. So absolutely, statement combinations A and D are correct. I hope the explanation is clear to everyone. Many short question. Yes, exactly, Antara. And nitrogen metabolism, I'll tell you, is very important for other state-based lecturership tests or competitive exams like SET, like West Bengal State, like Orissa SET, okay, set examination, they have a fetish for this chapter. So please prepare nitrogen metabolism very well. Okay, if I have skipped some questions, just a second, students. Oh, another question from Unit 6. After absorbing light, chlorophyll molecules in green plants exist in singlet and triplet states. Following are certain statements on singlet and triplet states of chlorophyll. So expect this question is from photosynthesis. Now, students, this CSIR, you will see they have this pattern nowadays. They have this notion of giving such type of questions from photosynthesis, like based on no excited state, singlet state. So it's an amalgamation of Unit 13. If you just look at the question very minutely, students, Unit 13 and Unit 6. And this is the trend of CSIR. Yes, Antara, you got it correct. So they love the chapter nitrogen metabolism, right? Yes. And it's a very nice chapter also. It's my personal favorite chapter as well. Okay. Next. So first statement says a singlet state is short-lived in comparison to triplet state. Thank you. Thank you so much, Mahima, for your nice feedback, dear. Thank you so much. Okay. A singlet state is short-lived comparison to triplet state. Singlet state contains electrons with antiparallel spins, while triplet state has electrons with parallel spins. Singlet state contains electrons with parallel spins, while triplet has electrons with antiparallel spins. So, students, can you, any guesses? Any guesses from here? Yes, Karthik, a very good evening. Any guesses, students? What can be the expected answer? Okay, it should be long lived. Yeah. It's a printing mistake. It should be long lived. Just a second. Yeah. So statement B is a singlet state is long lived. Okay, so I'll tell you. See, always a singlet state is short lived in comparison to triplet state every time. Now, exactly this portion has been given from a particular topic under photosynthesis, which is called as emission spectroscopy of chlorophyll. Emission spectroscopy of chlorophyll. Okay, and that is why I was telling you that this concept or this type of question is an amalgamation of the concept from Unit 13 and Unit 6, both. Okay. Yes, so Kritika, I have rectified the statement B. It is long lived. Okay. Now, so emission spectroscopy and we have Fret, Jablonski diagram. They are very important. So that part, okay, because there are there is a concept of energy transition. Okay, energy transition state from blue light spectrum to red light spectrum. So all these things comes under emission spectroscopy. So you need to understand the concept of spectroscopy very well from Unit 13, and you know about the chlorophyll concept from Unit 6. So that won't be a problem. Now I'll show you the explanation here somewhat. So singlet state is attributed to the appearance of a single spectral line. And this occurs because of all the electrons are paired. So originally the singlet state is referred to as a set of particles that had a net angular momentum of 0. That is, its spin quantum number is 0. And triplet state, on the other hand, indicates triple splitting of spectral lines. And moreover, students, this highly reactive singlet oxygen, it can also be produced via the triplet chlorophyll formation. So triplet is a very active state. Okay, and you can see also here, it's mentioned that triplet state, on the other hand, it happens due to the presence of two unpaired electrons. Okay, and these are the reasons. So the more we have the unpaired electrons, so it will have a much more powerful state or powerful condition. So we say that singlet state is very short lived in comparison to triplet state. Okay. And accordingly, singlet state will be containing electrons with anti-parallel spins, but triplet has got electrons with parallel spins. So because it has got two electrons, almost I said, so always they will be forming a parallel spin. Okay, and just remember this thing, students, very well. It's a universal characteristic, it's a universal nature that always, if this any a photon is having or it's existing in its single state, it will be bearing electrons. Like when we say single state of any particular element, say it's oxygen, whenever it is bearing electrons with anti-parallel spins, definitely its longevity, its power is going down. So this is a very no universal nature that I told you. So this question is very, very important, students. Very important. Okay. So option combinations will be A and C. Okay. So now we'll proceed. I hope this question was clear to every one of you. Okay. So now we are done with unit 6, and now we'll be moving on to unit 7. So that is what. And another very important thing is, students, always remember few tips from unit 6 preparation I want to share. That is, always stress on the locations. Okay, remembering locations from unit 6 is a very important strategy. Okay, remembering locations, that is, each and every location of the metabolic reaction is very important. Okay, pathways. Okay, whenever you are learning the pathways, the suppose there are a lot of metabolic pathways in unit 6. We have Krebs cycle, glycolysis, okay, C4 pathway, CAM pathway. Don't forget to memorize the enzyme names. Okay, and whenever you are remembering the enzyme names, also try to memorize the step names. Each and every step is different from each other. Like one step can be oxidative decarboxylation, another step can be oxidation only. So these things, another step can be substrate level phosphorylation. So these are very, very important things that needs to be remembered, and it is common for every metabolic pathways, students, every metabolic pathways. So you need to jot it down. Suppose if you are studying Krebs cycle, so jot it down how many steps of no oxidative decarboxylation is there, or in C3 pathway, how many steps of ATP utilization, or if any substrate level phosphorylation is there. So try to jot it down like that. Okay, so it is very, very, it's a very important thing that needs to be done. All right, yes. So now we are into unit 7. So unit 7 is also very important unit, and it's all about system physiology, animals. Okay, and quite a lot of important questions do come from here. And if we talk about the weightage or the number of questions that come from unit 6 or 7, so definitely the number is almost equal. The number of questions that come from plant and animal, it's almost equal. So we have to give the yes Mahima, reversible and irreversible steps as well. That is also a valid point, Mahima. Absolutely, you have to also remember the reversible and the irreversible steps. Okay, and when it comes to unit 7, unit 7, yes, the strategy would be somewhat different from unit 6. Okay, because unit 6, I would say, it's sometimes the questions that come, it's very direct from the pathway, but unit 7, it's all about animals and humans. So you need to understand the concept well. So I would say, after studying each and every concept, try to make a summary of the concept that you have learned, and in a flowchart manner. In case of plant, if you see, there are all cyclical pathways, so your work is 50% easier over there. But in case of animal, we have lesser number of pathway or metabolic reactions, but we have all the mechanisms, like digestion, circulation, all these mechanisms are there. So we need to focus on the mechanisms, and for that, the best option or the strategy is to follow flowchart. So every topic you are doing, no, every whatever important topics we have, just try to make a brief flowchart for every one of them. That will really make your work easier. So let's take up the first question here. If in a blood transfusion, type B donor blood is given to recipient having type A blood, the red blood cells of donor blood would agglutinate, but the recipient's RBCs would be least affected. Okay, Sachi, let's wait. Okay, so let's see what others has to say about this question. So these observations can be explained in the following statements. Yes. So now everyone can read the statements. So statement A, B, C, D. So you tell me, take some time, tell me which one should be the correct option combinations. You can try once. So Antara is saying 4. I'm not sure though. No, which are... Okay, this has been said as which of the above statements is or are incorrect. Yes. You can tell me both ways. Let's frame the question like this. You can tell me both which one can be incorrect and which one would be incorrect. Yes, I have written in the statements as correct, but fine. It should be, we can treat the question as both ways. So tell me which of the statements will be correct and which of the one will be incorrect. First, tell me the correct combinations. First, tell me the correct combinations. Okay. So if I talk about the incorrect combination, so correct one will be, yes, exactly. So A and B should be the correct statements. Just a second, students, it has gone away. Yes, I hope you're able to see the question now. Yeah. So A and B are correct. So why A and B should be correct, students? Because agglutinins in the recipient's plasma caused agglutination by binding with type B agglutinogens. And the agglutinins of donor blood was diluted in recipient's plasma membrane. So I'll explain you the thing. So this is B. Okay. So B will be donating blood to A, correct? So this B has got antigen B and antibody A, right? Yes. And this A, the recipient would be having antigen A and antibody B, correct? So now what will happen? So the recipient's antibody B will be attacking the antigen B of the donor. The antibody B of the recipient's blood would be attacking the antigen B of the donor, and that is how they will be forming the agglutination reaction. So definitely statements A and B should be correct and C and D should be incorrect, because agglutinins in the recipient's plasma caused agglutination. How? By binding with type B agglutinogens. That means the antibody B from recipient's blood will be binding to the antigen B, and thereby it will be forming agglutinogens. Okay, so the correct combinations would be A and B. Okay, sorry, only A. And the agglutins of the donor blood was diluted. No, this statement has got no valid point. So correct statement is only A, and the incorrect one is C and D. That is, low titer of antigen anti-A agglutins is the cause of low agglutinations. No, it is not because of the anti-A agglutinins that is why it is bringing about such thing. No, the reason is the antibody B in recipient's blood is binding with the antigen B of the donor's blood, and thereby it is bringing about the agglutination. Okay, so this is how statement C and D would be incorrect. Okay, so this is somewhat the overall explanation that I have tried to keep here. To avoid a transfusion reaction, donated blood must be compatible with the blood of the patient who is receiving the transfusion. Okay, so more specifically, the donated RBCs must lack the same ABO and RHD antigens that the patient's RBCs lack. Okay, so this is a very important explanation. Now, so let's move on to the next question. Yeah. The time taken for the arterial systole and diastole in a normal heart are TAS and TAD seconds. I hope you're able to see the question respectively. If ventricular systole take TVS seconds, then calculate the ventricular diastolic time. So here, several option combinations has been given to you. So what is this TAS, TAD, TVS? TAS means time a arterial systole. Okay. What we mean by TAD? TAD stands for time arterial diastole. And what is TVS? TVS is time ventricular systole. So there is a generalized calculation methodology that we need to follow for such important calculations. Still, I'll be showing you an explanation. We have to consider this explanation regarding a very important concept here, which is called as breath holding time, or BHT. What is this BHT? It is the time taken by the subject to hold his breath as long as he can. And during such voluntary breath holding, these tissues continue to utilize oxygen and liberate carbon dioxide. And therefore, during breath holding, arterial P oxygen PO2 level falls and PCO2 level rises. So that's the most important thing that happens. And moreover, the calculation that I was talking about is, always remember, the arterial systole and arterial diastole will always be equal to the ventricular systole and ventricular diastole. They will be always equal. So students, and the total concept is, or the total time that is being taken up for both this calculation is 0.8 second. Yes. So if we go back, that means what should be the ventricular diastolic time? So as we are saying that TAS plus TAD is equal to TVS plus TVD. That means how should I get the ventricular diastolic? It's simple. Then we have to subtract TVS from TAS plus TAD. That's the simple thing. As TAS plus TAD is equal to TVS plus TVD, so how am I going to get ventricular diastolic time? Simply we have to subtract TVS from TAS plus TAD. Yes, absolutely. Is this clear, students? Okay. I hope this is very easy, but you need to know the concept. That is what. And the total time taken is by both, it's 0.8 second. You can note it down. Yes, Mahima, correct. Very good. Now, so now moving on to the next question. Yes. The question says, in high altitude, a number of compensatory mechanisms operate over a period of time to increase the altitude tolerance in humans, that is called as acclimatization. So the following statements propose this compensatory changes. So this is about a very important fact, that is in high altitude, how like the number of compensatory mechanisms operate over a period of time so that the tolerance level is increased, and that is what we call as acclimatization regarding this. So what are the statements given over here? The initial increase of ventilation is relatively small in high altitude. The initial increase of ventilation is small, but the ventilation steadily increases over next few days. Red blood cell 2,3-DPG is increased. The blood pH becomes alkaline. The oxygen dissociation curve is shifted to the left, though there is no graphical representation given for this question, but you have to understand that. So that is why the dissociation curve is very important while you are studying this concept from Unit 7. And the pH of cerebrospinal fluid is further increased. So we need to choose the option with both incorrect statements. So, can anyone guess what can be the answer? It should be like, in this sense, correct means we have to choose the incorrect statements here. Okay. Very good. I could see answers coming in. So, absolutely correct. Those who have given the answer, correct answer, but incorrect statements. Okay. So, I mean to say the correct statements in the sense the incorrect ones that is asked in the question. And this is very confusing, students. Please follow the question. Out of excitement, we sometimes go for, okay, what are the correct statement combinations? And we go for the correct statement combinations that unfortunately becomes the incorrect one. So, yes. Answer key 4. That means this is the incorrect statement combinations D and E. That is, the oxygen dissociation curve is never shifted to the left, and the pH is never increased for cerebrospinal fluid. Correct. So, high altitude physiology can be divided into the study of short term changes, and when it is short term changes, it is with the exposure to, it's called as hypoxia or hypobaric hypoxia, that is acute response to hypoxia, and long term acclimatization. Two things can happen: short term changes, long term changes. Okay. So acute exposure to the ambient atmosphere at extreme altitude illness can lead to acute syndromes that affect lowland. Students, are you able to see the screen properly? It has enlarged, I guess. Just a second. Yeah. So, acclimatization is the set of beneficial processes whereby the low-land humans respond to a reduced inspired partial pressure of oxygen. And high-altitude illness can be divided into acute syndromes. Okay, so this is about the, no, we just they say high-altitude pulmonary edema, and also these are definitely the definite symptoms of high-altitude sickness. And regarding high-altitude sickness, these two statements, like definitely and never the cerebrospinal fluid will get increased or further increased in case of like, which we called as high altitude pulmonary edema, never it will happen. And the dissociation curve, no, it will never, like the shifting is never be towards the left hand side. Okay, so the incorrect combinations would be D and E. Now, so moving to the question, respiration can be inhibited voluntarily for some time, and the point at which the respiration cannot be voluntarily inhibited is known as a breaking point. Okay, so following explanations are offered for the breaking point. So what is this breaking point? Can anybody tell me? Yes, it is already mentioned that at which respiration cannot be inhibited. So it's a very vital thing, and breaking point is a very important concept, students. And based on it, you will see here the statement combinations that has been given. It's not related all for the breaking point. It has diverted no the concept. It has diverted from the concept being given here. Like the first two statements, if you look at J receptors stimulating respiratory centers, Herring Brewer reflex, it's not absolutely related to the breaking point concept. Okay. So yes, now based on this, actually I have gave you the hint only for the answer. So let's try. So which of the following statement combinations will be correct? Basically, the breaking point is reached when this alveolar partial oxygen stimulates the central and peripheral chemoreactors. Okay. So that is the major and initial thing that happens, but still, okay. So Antara is saying 3 or 4. Okay. Yes. So the correct option combinations include option 3. Okay. And that is R and S. That means the rise of arterial PCO2 stimulates the respiratory center, the rise of CO2 and the fall. Then only there will be a breaking point, right? When the respiration cannot be voluntarily inhibited, that is called as a no breaking point. So when it will trigger, when will it happen? The time is when there will be a rise in the CO2 level and when there will be a fall in the PO2 level. Then only these two conditions, if it is happening, then only it will be leading to this thing. And see, regarding this, we have no connection with the J receptors and Herring Brewer. So I have just kept a small concept on that. Yes. No, no, no. 3 and 4 are not same. Vijay, look at the statements, dear. The rise of arterial P CO2, when CO2 level is rising and fall of oxygen, then only this thing is happening or this thing will be happening. Okay, please read the statement. See, go through the statements very well. Got it? So, are they same? No. Okay, RNS. Yes, it should be Q and R. Yeah, oh, Q and R is there. P and Q, RNS. So, it can be P and S. Okay, but yes, this is the correct option combinations. Exactly, exactly. So see here, the juxtacapillary receptors or pulmonary C fiber receptors. Okay, that is the statement it was there. J receptors stimulate the respiratory centers. So they are sensory nerve endings located within the alveolar walls in juxtaposition to the pulmonary capillaries of the lung, and they are intervenated by fibers of the vagus nerve. And specifically, these J receptors, they respond to events like pulmonary edema, pulmonary emboli, pneumonia, all these things. And when it comes to Herring Brewer inflation reflex, it's a reflex, students. Okay, that is triggered to prevent the overinflation of the lung. So that is what it has got no direct relation with the breaking point, but still we can relate, but it has got no direct relation. But that is the thing, we have to connect with each and every small concepts that are part of, see, it's a part of respiration. So yes, when we talk about juxtacapillary receptors, it's a part, yes, it's a sensory nerve ending, but yes, it has got a connectivity with pulmonary capillaries. Okay, and they respond to such events like pulmonary edema, etc. And Herring Brewer is again a reflex that is triggered to prevent overinflation of the lung. Okay, yes, Kanis, hello. So that's all for today's session, students. It's time to end up. So before we end up, this is a big announcement for every one of us. We are starting with another batch of CSIR net, which is called as Lakshya batch. So for more information, definitely you are free to call these two numbers. And so whoever is enrolling, these are the benefits. That is, you are open to get Biotecnika's online study portal. There is 24/7 chat support and yes, coaching by CSI Net experts. So I would say for your better preparation, you can be a part of Biotecnika, okay, to shape your preparation for the upcoming June and December examinations. Yes, so students, if you have any other doubts, you can ask me. If you have any other queries from the unit 7 and 6. Okay, Antara, that's great. Thanks for this wonderful feedback you shared. Okay. Thank you so much, Devanshi. I hope this session will really benefit you, and the entire webinar series will be a really interesting and informative one for every one of you. Yes. Okay, so thank you so much, and my best wishes with every one of you. Yeah, thank you, Mahima, and all the best for your preparation for your upcoming exams. Thanks a lot.